Hi everyone, thank you all for being here. My name is Golia Shafiei and I'm a PhD student at the Montreal Neurological Institute at McGill University. Today I'm going to share with you some of the recent results of a recent project I've been working on. Here in this project, our main aim was to investigate the spatial patterning of tissue volume deformation in schizophrenia and its relationship with the underlying network architecture. As you may know, anatomical abnormalities in schizophrenia has been previously observed and reported. So some examples of these abnormalities include whole brain volume loss and enlarged ventricles, cortical thickness deficits, as well as volume loss in cortical and subcortical areas such as insula, anterior cingulate cortex, and hippocampus. Thank you. Sorry. In addition, white matter disconnectivity have been previously observed in schizophrenia. So basically, we can identify these local abnormalities or these brain areas that are abnormal, that have abnormal properties in patients compared to healthy controls, and we can construct these localized maps of abnormalities. So now, if we think of the brain as a complex interconnected network, as in any network, we can expect that pathology or dysfunction can spread among the linked elements. So this network and its topology may shape or may influence the onset, the expression, and the propagation of a disease. So this brings me to the question we try to answer in this project. Does the spatial patterning of volume loss in schizophrenia reflect the brain network architecture? So for the first step, in order to answer this question, we quantified volumetric differences in schizophrenia patients compared to healthy controls using T1 weighted structural MRI images obtained through the Northwestern University Schizophrenia Data and Software Tool, the NUSTAS database. We use deformation-based morphometry, or DBM, to estimate local changes in brain tissue volume density in the two groups compared to a group template. And we quantified brain tissue volume deformation in schizophrenia as the contrast between the DBM maps of the two groups. So the results are shown in this figure, where the warm colors show higher volume loss in patients compared to controls. So the next question we were interested in was to investigate whether these deformation patterns are focused to or are more pronounced in specific subnetworks or systems. And in order to answer this question, we first looked at the mean deformation values in the resting state functional networks defined by Thomas Yeo and his colleagues. We then compared these mean deformation values to a set of null distribution of means generated by a spatial permuting approach, or the spin tests, which basically preserve the spatial autocorrelation in the data, where we observed a significant volume loss in the ventral attention network. In addition, we investigated deformation relative to cytoarchitectonic classification of human cortex according to the famous von Economo atlas. And again, using the same test, we observe significant volume loss in insula and limbic cytoarchitectonic classes. So all these suggest that these deformation patterns observed in schizophrenia are more specific and are more pronounced in specific systems which have specific functional roles or cellular composition. So given the fact that these deformation patterns are more pronounced in specific networks, suggests that maybe these volume loss patterns that we observe in schizophrenia are conditioned by the underlying network architecture. So we tested this directly using structural and functional connectivity networks obtained through a healthy independent data set. So if this is the case, a node or a brain area which is connected to brain areas with high deformation or high volume loss is expected to have greater deformation or volume loss compared to a node which is connected to relatively healthy neighbors, which is expected to be relatively healthy as well. So we tested this hypothesis by looking at the relationship between the node deformation and the mean deformation of its connected neighbors, where we define the neighbors as the brain areas that are connected to a given node with an underlying structural connection. So looking at this relationship, we see a positive correlation between the two, confirming our hypothesis that that the amount of volume loss in a node is positively related to the amount of volume loss of its neighbors. We repeated the same analysis, looking at the functional connectivity and functionally defined neighbors, where we weight the deformation of the neighbors by the functional strength of the connections, and we observed the same positive relationship. In order to assess, see whether these results depend on parcellation resolution, we repeated the analysis in 5 different parcellation resolutions, and we saw the same relationship more or less between the node and neighbor atrophy. And in addition, to assess the extent to which these associations depends on the network topology and geometry, we constructed 2 sets of null models. In one, we randomly rewire the structural network, or we move around the edges while preserving the node degree, the degree distribution, and in the other one, we preserve the edge length distribution in addition to the node degree, so basically preserving both topology and geometry. And in both cases, we see that the relationship between the node-neighbor atrophy is significantly correlated, is significantly higher than what we observe in the null models. We repeated the same analysis, looking at the functional connectivity, where we randomly reassign the node time series, or we weight the randomized structural networks by the functional connections. And again, we see that the correlation in the empirical networks is significantly higher compared to the null models. So having seen all this and having seen that these deformation patterns are related to the connectivity networks, the next question we asked was whether we can identify an epicenter for the disease. So basically our hypothesis here was that if a node is highly deformed and if it's connected to the neighbors who also have high deformation, is more likely to be an epicenter compared to a node which is connected to relatively healthy neighbors. So in order to test this, we ranked the nodes based on their deformation values and their neighbors' deformation values, and we identified the nodes that were highly ranked in both lists, as shown on the brain surface here. And according to this analysis, the areas that were most likely to be an epicenter were located in cingulate cortex, which is somewhat consistent with the previous literature on involvement of ACC in schizophrenia. The final question we tried to answer here was to see if there is a relationship between these deformation patterns and gene expression. So this is likely to be the case given the literature on the genetic origins of schizophrenia. So in order to test this, we measured the correlation between the deformation values and the gene expression data obtained through Allen Human Brain Atlas. We constructed 2 ranked lists of genes according to how highly their expression were correlated or anti-correlated with the deformation patterns, and we submitted these ranked lists of genes to a gene ontology analysis tool, Gorilla tool as shown here, to identify Go terms that were highly overrepresented in our ranked list of genes. So according to this analysis, Go terms that were associated with greater deformation were related to biological processes such as nervous system development, synaptic signaling, and cell communication, which is somewhat consistent with what we saw before, where we saw that volume loss was related to larger scale connectivity, but this is more locally and in more maybe synaptic level. On the other hand, the biological processes that were associated with lower deformation had to do with immune response and inflammatory response or defense response, suggesting that maybe genes with more protective roles were overexpressed in these areas, were preferentially overexpressed in these areas. So in summary, what we show here, our analysis showed that the schizophrenia-related deformation is linked to structural and functional network organization of the brain. Tissue volume loss appears to be centered at brain regions associated with cingulate and insular areas. And the relationship may be shaped by transcriptional factors involved in signaling and immune function. And with that, I want to thank everyone in the lab, including my supervisor and my colleagues, our collaborators, specifically Dr. Dagger and Dr. Chakravarti, and the funding agencies. We also have a preprint available if you wanted further information, and I'd be happy to talk about this in more details during the poster reception right after this session. Thank you very much.